Kerry Bloom's lab is working with the NIH-sponsored SISM research team at the University of North Carolina to investigate the structure and forces in the metaphase mitotic spindle of yeast. This is when the pairs of chromosomes line up so that one copy of each will go to each daughter cell as the cell divides. The team proposed this geometric model for the chromosomes. There are 16 pairs of chromosomes, with one of each pair attached to the left side of the spindle and the other attached to the right side. Here we've colored the pairs differently to make their structure apparent. 4 of the pairs are drawn in each color. For visual clarity, we hide all but 4 of the chromosome pairs, revealing the proposed linear loop structure, with each chromosome having 2 loops so that there are a total of 4 loops across each pair. We're only showing the section of the chromosomes within the spindle and hiding the portion of each chromosome that exits the spindle region radially at the midplane between the 2 ends of the spindle. Shown here as white rings, a protein named cohesin attaches different chromosomes to each other. In this model, they keep the loops tied together and the chromosomes are free to slide through the rings. We know from experiments that the rings are located about 22 nanometers away from the center of the spindle, which is where they're shown here. Based on fluorescence microscopy experiments, we know that this model includes the correct lengths of chromosome material and the correct number of cohesin molecules arranged at the correct distance from the center. But this is just a geometric model. It does not include the effect of the forces we know are acting on the structure. In the actual cell, the chromosomes have a stiffness and resistance to bending, and there is thermal motion. Molecules are bouncing around and colliding with the chromosomes due to the heat in the system. To see whether the model still fits in the presence of such forces, we modified the open-source SOFA simulation environment to add thermal forces and bending rigidity. The regular structure of the rings is disrupted as molecules randomly collide with them. Collision detection in the simulation prevents the chromosomes from passing through each other, resulting in pressure as the chromosomes compete to fill up the volume around the spindle. The ends of the spindle are attached to microtubules, which are not displayed. This prevents the spindle from shrinking and stabilizes the basic structure. The thermal motion has caused the large loops to collapse inward as they become more tangled. At the same time, the pressure has caused the inner radius of the spindle to expand into a football-shaped structure. These motions counterbalance, leaving the cohesin rings at the same distance they were originally. If one or the other of these motions had dominated, there would have been a net change in cohesin position, invalidating the model. Here we watch the simulation from the beginning, seeing the reduction in outer loop radius and the increase in inner radius with little net motion of the cohesin. Our next step is to run the simulation longer to ensure that the chromosomes have the chance to explore the range of freedom that they have available to them in the real cell.